CD47 is a very interesting protein and was originally identified by many other investigators to have a role in regulating the innate immune system. In fact, it's a checkpoint for the innate immune system in that when it's present on cells, it can engage a receptor on phagocytic cells, macrophages or dendritic cells, and thereby prevent them from eating the target. So we think about CD47 as a don't eat me signal and a checkpoint inhibitor for the innate immune system. And now, almost 10 years ago, in work that was done when I was in Irv Weissman's lab at Stanford, we discovered CD47 to be a critical regulator of the phagocytosis of AML cells, and most importantly showed at that time that an antibody that could block CD47 had very potent activity at stimulating phagocytosis and elimination of the AML cells. Now, we developed a program at Stanford to bring a humanized antibody against CD47 into clinical trials, and through funding from the California Institute for Regenerative Medicine, CIRM or CIRM, we developed a humanized antibody while we were inside the university structure and brought it into 2 phase 1 clinical trials. One in AML patients that has been underway as a phase 1 study in the UK with colleagues collaborating from the AML working group there. And a second trial at Stanford initially studying phase 1 activity and safety in patients with solid tumors. That was a parallel line of investigation. Now, in full disclosure, in 2015, we launched a new startup company, and in 2016, that company, 47 Inc., took over the program with licenses from Stanford University, and I'm involved in that as a consultant and a founder. But the program has advanced quite a lot. The antibody that we developed, called HU5F9G4, is now in 5 clinical trials across a number of indications, some in monotherapy, some in combinations. A couple of those trials are in leukemia, but the others are in solid tumors. So a lot has been done to define the safety of this particular approach, and I anticipate in the next year we'll start talking about preliminary efficacy results coming from these studies.